Since we were talking about T cells, let's move toward one important point, the point in which we will be focusing about the relation between T cell and B cell. Now let's find out what is that. Okay, since we have talked about a lot of CD molecules that are present on the surface of B cell and T cells, now I would like to take you toward the important concept in which we will know about the interaction between B cell and T cell. Okay, let us suppose this is our B cell. Okay, let me just make you clear. Let us suppose this is our B cell. What is this? This is our B cell. Okay, this B cell consists of what? BCR, B cell receptor on the surface. And what this B cell receptor does is, this B cell receptor recognizes or it receives antigen. What is this? Okay, let me draw it with a brighter color. Okay, this one. And here this BCR is receiving what? Antigen. Okay, this is antigen. This is antigen. And this is BCR or B cell receptor. That's all. Now, okay, we have learned this much. And another thing that we need to know right here is, there is one CD molecule on the surface of B cell, and the name of that CD molecule is CD40. What I said? CD40 molecule. Okay, and while learning about this CD40 molecules, we just were mentioning about CD40 ligand, which was present on the surface of T cell, if you remember. Okay, let's draw now the T cell. Okay, this one is our T cell. Okay, as we have recently mentioned that this CD40 is combining with, or it is binding with our the CD40 ligand, and here it is CD40 ligand. Here these two are combining, or these two are accompanying each other. Okay, that's all. Now another thing, okay, let's draw, it is the nucleus of T cell. Now another thing, this antigen which was presented on the surface of B cell is now going to be presented on the surface of T cell with the help of what? With the help of receptor, and this is T cell receptor. What is this? This is T cell receptor. Now this T cell receptor, okay, this is how antigen is presented to T cell. And here this antigen, obviously it is what type of antigen? It is proteinaceous antigen. It is not non-proteinaceous antigen. Proteinaceous antigen requires the support of T cell to produce antigen. Okay, now as this antigen is presented to T cell, right, let's write it here, it is proteinaceous antigen. Okay, since this antigen is presented to T cell receptor, this T cell will get activated. As this T cell will get activated, there will be the production of what? Antibody. Now what is that antibody? That antibody is IgM. Okay, now that antibody is IgM. Okay, we have already talked about IgM. What is IgM? IgM is the first antibody produced, or it is the antibody of primary immune response. That's all. Okay, now here T cell is activated. As T cell is activated, there comes the production of IgM. IgM is produced. Now what about other types of antibody? I mean, how they are produced? I mean, there are many of others like IgAMDE. I mean, IgG, IgA, IgM, we have already mentioned it here. IgD, IgE. How they are produced? They are produced when this IgM will be acted by some other chemicals. Okay, what are they? Okay, the important thing that we missed we missed it knowingly, is that CD40 and CD40 ligand, this interaction helps in formation of interleukin 4 and interleukin 5. Now why are these important? These are important because this interleukin 4 and 5 plays important role in converting this IgM into other type of antibodies, such as what? Such as IgG, IgA, IgE. Okay, that means here we are talking about converting IgM into other type of antibody. What is this process called? This process is called isotype class switching. We can say isotype switching, or we can say class switching. So let's mention it here. What I said is, this is called what? Isotype switching or class switching. Both are the similar thing. That means initially the antibody that we get is IgM, and with the help of interleukin 4 and 5 they are converted into other type of antibodies. Here we can see is, now this interleukin 4 and 5, they came from where? They came from this CD40 and CD40 ligand interaction. And what is this CD40? It is the part of B cell. And what is this CD40 ligand? It is the part of T cell. That's all. So right now we have learned about isotype class switching, isotype or class switching, and that is what we need to know about T cells. That's all. Now after this topic we will proceed toward another topic, and another topic is NK cells. So see you at the next lecture. If I can figure it out, you too can figure it out. With that motive, let's begin with another topic of immunity, and that is natural killer cell, or we can simply say that is NK cell, right? NK cell. Okay, before getting into its deep points, let's know what is natural killer cells. We have mentioned earlier in earlier lecture also that, let's revise it again. The natural killer cells are the part of what? These are the part of innate immunity. And if we recall our concept on hemiopoiesis, then we will remember that there was what? There was lymphoid stem cell. What was there? Lymphoblast. Let's say lymphoblast. And this lymphoblast was converting into what? T cell, one into B cell, and NK or natural killer cells. The one that were going into thymus were called T cell. The one that were going into bone marrow and were maturing there were called B cell. And the one that will be naive, or that will not be converted into any other form, were called natural killer cells. These are the naive one. I mean the one that are not converted or that are not changed into any other form. These were the natural killer cells. When we recall more about our previous lecture, we will remember that T cell and B cell were the part of adaptive immunity. And the NK cell was a part of innate immunity. Okay, now NK cell. Let's get deep into NK cells. What are the NK cells? Obviously the NK cells are the component of innate immunity. And these NK cells, the important feature of this NK cell is they secrete cytokines, same like of T cells. What I said, I mean these NK cells secrete cytokines same as T cell. Then what differentiates cytokines and T cells if they are functionally similar? Then the thing that differentiates then what point differentiates this NK cell and T cell? The point, because since we have said that they secrete same type of cytokines, then what differentiates them? The thing that differentiates them is, it is all about TCR. That means T cell receptor are only present in T cells, but these T cell receptor are not present in NK cells, and that is what it differentiates from T cells. Okay. Another important point that we need to know is earlier, previous some years ago, these NK cells were called large granular lymphocytes. What I said, large granular lymphocytes. Okay, these were called large granular lymphocytes. Why they were called large granular lymphocytes? Obviously because they were larger in size, and we said that these NK cells are the cells which are naive in nature, or which don't change their form. And we have already mentioned before, these are naive. That means they are like what? Lymphoblast. So lymphoblast means they are like lymphocytes. That's all. We can get this point through that concept also. Okay, and NK cell, what is its function? Its function is similar. Its function, through the name we can know what is its function. Its function is it kills infected cells, viruses, tumor cells. That means it kills infected cells, tumor cell, and viruses. That is all. Okay, now we have talked about brief introduction about NK cells. Now let's draw one NK cells and let's talk about its feature in detail. Let us suppose this is our, this is a bit thicker. Let us take, this is our NK cell. This is our NK cells. The important feature of NK cell is it consists of two type of receptors. Okay, what are the two types of receptors? Okay, let's draw nucleus here. The two type of receptors are, the one receptor is here and another receptor is here. The receptor that I'm showing in green color is called the inhibitory receptor. And the one that I'm showing in yellow is what? Obviously stimulatory receptor. Okay, these are the two receptors found on the surface of NK cells. Obviously these stimulatory cells will activate NK cell, and this inhibitory will inhibit NK cell. This will inhibit NK cell. Now, while talking about this thing, we will know an important fact, and that important fact is there is molecule called MHC. What I said is there is molecule called MHC. What is the full form of MHC? Is major histocompatibility complex. Major histocompatibility complex. And what is the important point about it? The MHC molecule is found in every cell of human body. Almost all normal cell of human body. It is present in almost all normal cells. And another point that we need to know about MHC is, one point we already mentioned, it is present in almost all normal cell of human body. And another point that we need to mention is there are some exceptions about where they are not present, and we will be talking about them in MHC because there is separate lecture on MHC also. Okay, and another point about MHC molecule is this MHC molecule binds to inhibitory receptor of NK cells. Inhibitory receptor in MHC. When it binds to inhibitory receptor, obviously that means here MHC is acting here. As it acts in inhibitory receptor, obviously it will cause the inhibition of NK cells, or NK cells will be inhibited. And if NK cell will be inhibited, then obviously it will not perform the function of killing bacteria, viruses, tumor cells, cancerous cells. Okay, we can just interpret our one important concept through this. Okay, there are several cells in my body, normal cells in my body. These normal cells consist of MHC and NK cell, and the behavior of NK cells is it kills cells, cancerous cells. We can we can understand like this: when cancer cells, when NK cell come in contact with normal human cell, then at that time, why it does not kill that normal cell? It does not kill that normal cell because that normal cell consists of what? MHC. And what MHC does is it binds to inhibitory receptor in inhibitory receptor in NK cell. It binds to inhibitory receptor in NK cell, and if it binds to inhibitory receptor in NK cells, then will NK cell be activated? Obviously not. That's why NK cell won't affect normal cell of the human body. Okay, what in the case of virus, or what in the case of virus infected cells or cancerous cells or tumor cells? Okay, in the case of such infected cells, what happens? In the case of virus infected cells, or okay, I'll write here virus infected cell, or we can say, what we can say is mutated cells. Okay, in these cells what happens is, in these cells there is less expression of MHC cells. That means in this case, or in these cells, we will find MHC cells in less amount. I mean, we will find expression of MHC in less amount. Less expression of MHC. Okay. If there will be less expression of MHC, then which will be activated? Inhibitory receptor or stimulatory receptor? Obviously, MHC was activating. Okay, MHC was going and binding with inhibitory receptors. But here we are saying MHC is less in number. That means NHC is less in number, means there will be obviously what? Stimulatory function. There obviously will be stimulatory. There will be obviously activation of stimulatory receptor that will cause activation of NK cell. That means in this case there will be less expression of MHC. That's why the virus infected cells are killed. Virus infected cells are killed. That's all. That is how that is how NK cell works. Okay, now while talking about this thing, now we will mention some other important things here, and some of the important things that we are going to mention here is, okay, let's draw another diagram of NK cell. That means we have drawn it with pink color. I'll take pink color, and again I will draw one NK cell. I'll draw one NK cells. Okay, as T cell and B cell, there are several CD molecules on the surface of NK cell also. Okay, what are the CD molecules that are on the surface of NK cells? Okay, initially let me put here one CD cell, and this one CD cell is CD56, right, on the surface of NK cells, and another CD molecule on the surface of NK cell is CD16. And this CD16 is also called Fc receptor. Why it is called Fc receptor? It is called Fc receptor because it attaches itself, or it joins with the Fc portion of antibody, the one that we have learned in B cells topic or B cell lecture. Okay, that means what this CD56 does. Okay, we'll talk about that thing later on, but let's focus here. CD16. Let us assume that there is antigen, and this antigen is coated by what? This is coated by antibody, and this is the Fc end of antibody that you can see right here. This is the Fc end of antibody. This is antigen that I'm showing here. This is antigen, this is antibody, and this is the Fc end of antibody. This Fc end of antibody will bind with CD16 molecule that is present on the surface of natural killer cell. Okay, that means here there is these two things bind, and as these two things bind, this binding also causes what? The activation of NK cells. activation of NK cells. Okay, let me just, okay, this causes activation of NK cells. Okay, let me write it in simple one line. Okay, what you said is Fc portion of IgG. I'm writing Fc portion of IgG because the antibody that is usually here is IgG. The Fc portion of IgG combines with what? CD16, or it is also called Fc receptor. These two will combine and these two will cause what? It will cause activation of NK cell. And when these NK cells are activated, then there occurs ADCC. Now what is this ADCC? This is antibody dependent cell mediated cytotoxicity. What I said, okay, antibody, right? Antibody dependent cell, okay, cell mediated cytotoxicity. Okay, that's all. That means, okay, let's go through each word. Cytotoxicity, okay, that is done with the help of antibody. That means here there was antibody, and with this help of antibody, we are activating NK cells, and these NK cells is becoming active. That is what it is. Okay, now we simply are into the topic of NK cells, natural killer cells, NK cells. Okay, now moving further into further points of NK cells. That's all. NK cells, and this is all about NK cells. That's all about NK cells. Okay, okay. If I can figure it out, you too can figure it out. With simple motive, let's begin the next lecture on immune system activation. Especially, it is the basic concept of immune system activation. Okay, I'm writing here immune system activation. Okay, talking about immune system activation, initially let us go toward the basic concepts. And the basic concept, okay, let us suppose this is our human skin. This is our human skin, obviously the barrier, and there are several microorganisms in environment which are the antigen. Okay, these are antigens. Let me draw with different color. Okay, these are antigens, and these antigens are in different form: microorganism, viruses, bacteria, and these are present in environment. They can't enter inside our human body. This is all our human body. and here we have drawn, what we have drawn? We have drawn barrier which is skin. Okay, this antigen cannot enter in our body, but if there will be any problem, and there will be any problem and this antigen get chance to get inside the body, then initially they come in contact with antigen presenting cell. What I said is they will be taken up by APCs. What are APCs? Antigen presenting cell, APCs. These antigen will be taken by APCs, antigen presenting cells. Now what this APC does? Okay, we are talking here, whatever we are talking right now, it is inside the layer of skin. We are talking beneath skin. Okay, what these APC does is these APC will fragment these antigen into smaller peptides. That means they will break down these antigen into smaller peptides, and after breaking down into smaller peptides, they will express these antigen on the surface of on their surface. So these antigen will be now expressed on the surface. These small peptides of antigen will be expressed on the surface with the help of what? With the help of MHC molecule. Okay, and when they express these small peptides on their surface, they move from skin layer to circulation. That means now they go to circulation, and while going into circulation, I mean when they are passing or circulating in human body, at that time they reach into lymphoid organs. And in that lymphoid organs there will be concentration of T cells. That means circulation, they reach where? Lymphoid organ, and in that lymphoid organ we will find what? T cell. Now what happens is these APCs, antigen presenting cell, and T cell will interact with each other. Okay, such as this was our what? APC. This was our APC which was expressing peptides, small peptides on their surface. Now these will present these small portion of antigen to our what? T cells, which are on the lymphoid organ. That means this is our T cell, and here we can see this is T cell receptor. We can see here this is, okay, this is our T cell, this is our APC cell, and this molecule is MHC, this is TCR. And that is how these antigen are exposed or are presented to T cell, and after this the T cell become active. Okay, whatever we said here, we have drawn this simple diagram, and in this simple diagram I would like to again tell you, I like to recall all the points. Initially what happened? Initially entry of microorganism, and in point 2 what we came to know is, in point 2, phagocytosis of pathogen by APC. And here, okay, I'll just mention here the entry of antigen was taking place, and here in this place these APCs were engulfing or phagocyting these antigen, and after phagocytosis these antigen were expressed on the surface. These were expressed on the surface of APC, and these expressed the APC that were expressing antigen on the surface were moving into circulation. And they were moving into circulation. When moving into circulation, they come and they pass through lymphoid organ, and where they meet T cells. And while meeting T cells, T cells, they activate T cells. The T cells are activated. And this activation of T cell, okay, this activation of T cell will lead into what? Lead in the production of effector T cell and memory T cell. Effector T cell and another is memory T cell. Now what are these effector T cells? Okay, natural killer cells, right? No, immune system activation. We are into immune system activation now. What are these effector T cells and memory T cells? The effector T cells are the T cells that will provide its action at the time of infection. That means deals with what? Present infection. And the memory T cell deals with later infection, obviously later infection. Okay, now we have got the basic concept of how it works. This is how it works. Now, okay, here the thing that we were talking a lot about is APCs. Now let us know a detail about APCs. What are APCs? APCs are antigen presenting cell. Okay, what we're going to learn right now is about APCs. What are these? Antigen presenting cell. Antigen presenting cells. Okay, talking about antigen presenting cells, I'd like to draw one antigen presenting cell that we have already mentioned above. Okay, this is our APC. This is the nucleus of APC. As we have already mentioned, there was one MHC on its surface that was expressing what? Antigens. That was expressing antigen to what? To T cell receptor, and this T cell receptor were present on the surface of what? T cells. These were present on the surface of T cells. Now, in chapter T cell, we have learned that T cell, okay, this is APC, and here it is T cell. In the chapter T cells, we have learned that there are two types of T cells. What are the two types of T cells? One is CD4 T cell, and another is CD8 T cell. Right? One is CD4 T cell and another is CD8 T cells. What was the name of CD4 T cells? It was called helper. If you remember, it was called helper. And CD8 is cytotoxic. Okay. Now we have already said that APC will present this antigen to T cell, and then T cell will be activated. Which T cell? The question is which T cell will be activated? Either CD4 or CD8. And for this, we need to know what was the class of MHC that was expressing antigen to T cell. And there are several type of MHC, 1, 2 and 3. Okay, what was the class of MHC that was expressing antigen to T cell? According to that, we will know either it will be CD4 or that will be CD8. Okay, that means we need to know about the different types of MHC, different class of MHC, let's say. Okay, if this MHC, I've already said you that MHC is a separate topic. We'll talk in detail about MHC in MHC topic only. Okay, that means this MHC, right now let's talk about two type of MHC. One is MHC1 and another is MHC2. Okay, if MHC1, if this antigen was presented by or was attached with MHC1, then at that time there will be a activation of CD8 T cell. And if in this MHC there was MHC class 2, then there will be a activation of CD4 T cell. It's simple, it's easy to memorize. Okay, how? Let me take you toward one fact. While talking about we're talking about CD8 T cell and CD4 T cell, we get the ratio of 1:2. I mean, the concentration of CD8 to CD4 is 1:2 in our body, and this is the factual data. And from this factual data, we can we can memorize that CD8 T cell will be activated when there will be Mhc class 1, and CD4 T cell will be activated when there will be Mhc class 2. From this fact, we can memorize it. Okay, now we were talking about what? We were talking about immune system activation. At the time we talked about APC, and we are right now talking about APC. While talking about APC, we are getting MHC. We are talking about MHC a little bit in brief, but let us talk a bit more about MHC. I already said you MHC will be taught in separate topic, but here we'll talk about MHC function. MHC functions. Talking about the function of MHC, the first function is it let pathogenic antigen to be expressed on the surface of APC, as we have done here. I mean, this MHC molecule, what it was doing, it was helping these antigens to be expressed on the surface of APC. That's all. That is the one function. I mean what one function is? Antigen. It helps antigen, right? Antigen to be expressed on surface of what? On surface of APC. That's all. And another one we have already talked about, the MHC function. I mean the MHC1. What it does, it activates CD8 T cell. Similarly, MHC2, what it does, it activates CD4 T cell. That is all, right? And from this point, we can just bring some other points. And what are the some other points? And these points are frequently asked in MCQs. So CD8 cell, we can say that from this point we can say CD8 T cell. What are these? These are Mhc class 1 restricted cell. That means they will be activated only if what? Only if Mh1 is present with antigen. Similarly, CD4 T cell. CD4 T cell are what? Mhc class 2 restricted cell. That's all. And what about NK cell? Okay, if you remember the fact, in NK cells we have also mentioned about MHC molecules. At that time we have said that our normal cells of our body, maximum normal cells of our body consist of MHC, and that MHC was, what was that? That was going and attaching with inhibitory receptor. Okay, if you recall the concept, we have learned in NK cells. I mean, we have learned in NK cells, natural killer cells. Yes, you can see here. We can see here, look, here we have talked about MHC. So we have said that CD8 cell, I mean CD8 T cell are Mh1 restricted cell. Similarly, CD4 cells are Mhc class 2 restricted cell. Then what about NK cells? NK cells are not Mhc restricted cells, because at that time where we were talking about Mhc, at that time we were not talking about any of the type of Mhc. So NK cells are not Mhc restricted. That's all. So we learned about these things, right? Okay, let us push ourselves a bit further. We're talking about the different point of MHC. So talking about a deal about MHC, the thing that we need to know is about MH1 and MH2 class of MHCs. Okay, let's say this is one MH1 and this is MH2. Okay, I'm writing here MHC and MHC type 2. Okay, we're talking about MHC class 1. Okay, I've already said you that this helps antigen to be expressed on the surface of APC. Okay, that means here we can draw one thing, and that thing is just this. Okay, don't worry. This is, what is this? I'm going to say you soon. Okay, and then this is our antigen. This is our antigen. The point where this antigen go and sits into MHC, that means this place go and sits into MHC, it's called antigen binding cleft. What I said is this point, okay, where this antigen go and sit, antigen where point where antigen goes inside what? MHC. It's called what? It's called, what I said is antigen binding cleft. Okay, I can just demonstrate right here. Okay, this is the cleft, antigen binding cleft, and this is the antigen, and we are talking about this one right now. Okay, in the case of MHC class 1, this antigen binding cleft is made up of what? That is the question. And this green one, the green one that you are seeing right here, this is alpha 2. This is alpha 2 chain, and the red one that you are watching right here is alpha 1 chain, right? Alpha 1 chain. And the blue one that you are looking right here is, I think it is not properly visual, but I'm trying to show it so well. And this blue one, it is beta 2. In the case of MHC class 1, this antigen binding cleft is made up of what? It is made up of alpha 1 and alpha 2 chains. These two, alpha 1 chain and alpha 2 chain. These two makes antigen binding cleft in the case of MHC1. And since these two chains are far from each other, they are also called what? They are called distal alpha chains. So we can say in MHC class 1, ABC or antigen binding cleft, ABC antigen binding cleft are made up of alpha 1 and alpha 2 chain, or we can say distal alpha chains. What I said here is distal alpha chains. That's all. Okay, now talking about MH2 type, and in this MH2 type, let me draw here the same, what? ABC, right? Antigen binding cleft, and where here in this case we can see okay, this one, and then we can see this one, and here what is this is beta 1, and the one that I'm showing here, it is alpha 1, and this is that I'm showing is alpha 2, and this is beta 2. Similarly, here we can see we have drawn what? One antigen, and this antigen where it is going into MHC is what? APC, antigen binding cleft. And here in the case of MHC, we can see that this ABC is made up of alpha 1 and beta 1 chain. And since these are also far from each other, we can say alpha, okay, we can say alpha and beta distal chains. That is the difference between MHC class 1 and MHC class 2. Okay, we started the topic on basic activation of immune system, and we are right now into MHC. We came into this topic. Why we came into this topic? Because in APC there were what? There were MHC molecules. That's why we provided a brief introduction to MHC. Now let's talk further about the APCs. What are APCs? We have already told that what are the APCs. Now going into the types of APC, there are different types of APC. Now let's proceed to our types of APC. Okay, talking about the type of APC. Okay, there are how many type of APC? Broadly, there are two type of APC only. What are they? They are professional APC, and the other one is, okay, let's write it this way. Other one is obviously non-professional APC. What are the professional APC? Professional APC, through the name we can say that professional, that means the one that should be called first, and non-professional means the one that should be called later on. The first work should be done by professional peoples, and then comes the role of non-professional peoples. So what are these professional APCs? Professional APCs are the APC that contains the largest concentration of MHC. That means that contains a largest concentration of MHC. As they contains a largest concentration of MHC, they have a lot of site for expressing antigen. That's all. And non-professional are the one that has lower concentration of MHC. Right. Talking about the professional APC, we need to talk about dendritic cell. DENDRITIC, dendritic cells. These are the professional APCs. What are dendritic cells? What are dendritic cells? These are professional APCs, and where are they found? They are found in skin, right? In skin and interstitial tissues. These are present in skin and in interstitial tissues in the form of what? In the form of Langerhans cells. What I said is LANGERHANS, Langerhans cells, and these are the most important. Why are these most important? Because these are the first thing that prevents antigen to enter in our body. So dendritic cells, they are present in skin and interstitial tissues, and they are called Langerhans cells. And talking about another site where they are present are lymph node and spleen. What I said? Lymph node and spleen. Talking about lymph node and spleen, they are present there in a form of follicle, so they are called follicular dendritic cells. Follicular dendritic cell. Okay, and this is important to mention that these follicular dendritic cells also act as reservoir for HIV. That's all. We have talked about professional APCs. About dendritic cells, and similarly under professional APCs there comes B cell. We have talked about B cells separately in one lecture, and talking about professional APC, we should not forget to mention macrophages. macrophages. These were all about professional APCs. Now what about non-professional APCs? Okay, let me highlight here. So that these were the professional APCs. Now talking about non-professional APC. Talking about non-professional APC, the one are fibroblast. 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 Similarly, endothelial cells. Endothelial cells. Thymic epithelial cell. Okay, these are non-professional APCs. And similarly, okay, we can understand with the basic concept here. In the case, normally physicians are there to treat a patient, but in the case of emergency, if large number of patients are there in which physician can't look after each of them, then at that time physicians need to take help of other staffs also. So the physicians are under the professional APCs, whereas the other are under non-professional APCs. That is what the difference is. Okay, now simply let's talk about the activation and inhibition of T cells. Talking about the activation and inhibition of T cells. Okay, activation and inhibition of T cell. Talking about the activation and inhibition of T cells, there is what? Antigen, and these antigen enters body. And when these antigen enters into body, they are presented by what? APCs. And these APCs express these antigen on their surface. Okay, now again the same drawing that we have drawn earlier. I would like you to take you toward this concept. This is our APC, right? And this is our T cell. Okay, this is our T cell, and this is our, okay, I'll write it here, APC. Now what happens is we have got here Mhc. Let me draw with it. We have got here Mhc, and this Mhc is presenting a lot of what? This Mhc is presenting a lot of antigens. Now here, these are presented also to what? The T cell receptors that we can see here, right? Okay, just let me write it down. These are antigens, right? These are Mhc, right? And these are T cell receptors. Let me zoom it. T cell receptors. Now the signal or the signal that is sent to T cell for its activation through these things, or we can say the signal that T cell receives while there is an interaction between Mhc, antigen and TCR. This signal is known as signal 1. What I said? Signal 1. For T cell to activate, only signal 1 is not sufficient. That means you are starting bike, okay, motorbike, and in that motorbike you initially what you do is you keep keys, right? You insert key. Just by inserting key, the bike will not start, right? To start that bike, now the next thing that we need to do is either we need to press the self start or we need to kick it, right? And then only the bike starts. That means the process in which we are keeping keys into that motorcycle, that is signal 1. And the next step, or when we press self start or when we kick that bike, and that is the next signal, and that signal is co-stimulatory signal. That means both the signal are important for the activation of T cells. What is that another one? We say this is signal 1, and another one is co-stimulatory, co-stimulatory signal. And how is this co-stimulatory signal produced? Okay, if you remember in T cells we have learned about CD28 molecules that was expressing on the surface of what? On the surface of T cells. That means there was CD28 molecule. What is this? CD28 molecules, and it was interacting with our one and only what? With our one and only molecule on the surface of APC, and that is B7. That means these two molecules were interacting each other, and the interaction between these two molecules is called what? Co-stimulatory signal. And both of these signals are important for the proper activation of T cell. If there will be only signal 1, then there won't be a proper activation of T cells. So for activation of T cells, we need to have co-stimulatory signal also. So the another thing that I would like you to take toward is external antigen. What this external antigen does is these external antigen provides signal 1, which was this signal 1, and also co-stimulatory signal. I mean, both of these were provided by external antigen, right? Antigen that comes from outside. What about self antigen? Self antigen means within ourselves. Okay, we're talking about, okay, I said you something about external antigen. External antigen. What I said is external antigen provides signal 1 and also what? And also co-stimulatory signal. That was all about external antigen. Now talking about self antigen. What is self antigen? Talking about self antigen, this self antigen provides signal 1, but it does not provide co-stimulatory signal. Okay, that means it provides signal 1 but no co-stimulatory signal, right? Okay, this was all about self antigen and external antigen. Okay, let's talk of more about self antigen. Okay, it was not providing co-stimulatory signal. That means, will there be activation of T cell? Obviously no, as we have already mentioned that for activation of T cell we require both signal 1 and co-stimulatory signal. Here there won't be what? There won't be activation of T cell, right? If there won't be activation of T cell, then what is the condition called? This is called T cell energy. T cell energy. Okay, what is T cell energy? That means the condition in which there won't be the activation of T cell. The condition in which there won't be T cell activation. That is what the T cell energy. That means T cell energy occurs at what time? At the time when there will be the presence of self antigen. Why? Because there won't be co-stimulatory signal. That's all. Okay, talking about these things. Okay, what is the importance of this? I mean, self antigen. What is the importance of self antigen? It is important because it helps to prevent autoimmune disease. Prevention of autoimmune disease. That's all, right? Okay. Autoimmune disease. And this was all about what? This was all about what? This was all about the basic of immune system activation, right? That's all for the basis of immune system activation. Now for the class, it will be of Mhc. So let's end it right here. Thank you. If I can figure it out, you too can figure it out. So with that simple statement, let's begin our topic with MHC. Now what is this MHC? Okay, MHC. Now we'll be talking about MHC. What is this MHC? These MHC are, while talking about MHC, we are talking about the group of genes. And genes, where are these genes present? The genes that are present in cluster in chromosome number 16. That means in cluster in chromosome number 16, right? MHC molecules, what are these? These are the cluster of genes chromosome number 6. Where exactly in chromosome number 6? In short arms. Okay, since these were for the first time discovered in leukocyte, they are also called human leukocyte antigen. What I said is, these MHC were first time discovered from what? They were first time discovered from what? From leukocyte. So these are called what? Human leukocyte antigen. Human leukocyte antigen, right? Or we can say it HLA also. Okay, we're talking right now about MHC, major histocompatibility complex. Okay, I forgot to tell you full form of it. It is what? Major histocompatibility complex. Okay, well, proceeding through its content, the another important thing that we need to know about is its type. That means the group of genes that are forming MHCs. Okay, let me draw one simple diagram, and the diagram goes like this. Okay, here I'm going to show different group of gene. Okay, the first one that I'm showing here is, okay, let me draw it like this, and then let me draw it like this. Okay, the first thing that I'm going to write is, I'm going to write the first group of gene, that means group 1 gene. What I'm writing here is, these are group 1 genes. Talking about group 1 genes, it again further contains different portions. The portions such as, let me show you here, it contains certain portions. And what are these portions? These portions are A, B and C. What? This one is A, this one is B, and this one is C. What are these ABCs? These are the portion of group I gene, and what we can say them, we can say HLA-A, we can say HLA-A, what we are saying, HLA-B and HLA-C. So this was the group I genes. And in middle, you might be thinking there would be group 2 genes, but no, there is the presence of group 3 genes. And at last we have group 2 gene. Okay, as of group 1 gene, here also we can see the different portion of group 2 gene, right? Okay, and the different portion of group 2 gene are what? Okay, let me mention here the different portion are, we can see DP portion, DQ portion and DR portion, right? And these are the portions of group 2 genes. Okay, these are the genes. Okay, talking about group I genes. While talking about group I gene, the thing that we need to know is these are responsible for giving MHC class 1 molecules. I mean, they give what? MHC class 1 molecule, right? And what about this group second genes? These are responsible for giving MHC class 2 type, right? And what about the third group? These don't give MHC class 3. There is nothing called MHC class 3. So what it does then? The thing that it does is it helps in producing different type of cytokines, or it helps in producing different type of protein. It codes for C2, right, C4, or also we can say it codes for prophylactics. And another thing that it does is it also codes for tumor necrotic factor alpha. That is the importance of group 3 genes. Okay, what was group 1 gene doing? It was responsible for giving MHC class 1. And now what the MHC class 1 does? Okay, talking about MHC class 1, there are important points that we need to know. MHC class 1 are present in all nucleated cell. What I'm saying, present in all nucleated cells. The non-nucleated cells that we can remember is RBC and platelets. That means these are not present there, but that is a wrong statement, because they are also present in platelets, even though platelet is what? Non-nucleated. Okay, but it is not present in RBC. And another point that we need to know is it activates MHC class 1 activates which type of T cells? It activates CD/T type of T cells or cytotoxic. We have already learned about this thing in T cell, right? Okay. And another point that we need to know about MHC class 1 is, just highlight this, the one in MHC class 1, and how we can detect MHC class 1. We can detect MHC class 1 with the help of allo antiserum. What I wrote is allo antiserum. That is how we can identify or we can detect MHC class 1. Now talking about MHC class 2. These MHC class 2 are present where? These are present in all antigen presenting cells. Okay, where are they present? They are present in all APCs. Talking about APCs, what are the types of APCs? Obviously, non-professional and the professional. And these things we have learned in detail in what? In basics of immune activations. Okay, APCs, non-professional and professional APCs. Now, what is the role of MHC class 2? What it does is it activates CD4 type of T cell. And for the talking, how can we detect MHC class 2 molecules? We can detect MHC class 2 molecules with the help of mixed leukocyte reaction. What I say is mixed leukocyte reaction. Okay, this was about MHC1 and MHC2 class of MHCs. Now, talking about the uses of MHCs, right? What are the uses of MHCs? Talking about the uses. Well, talking about the uses, the first thing that we need to talk about is it is used in genetic association of disease. To know about the genetic association of disease, what is that thing? Okay, genetic association of disease. Talking about genetic association of disease, the point that we need to know is where there are the presence of HLA, right? What we are talking about here, where there are the presence of HLA DR3 or DR4 molecules, these are associated with, or there is the high chance of occurrence of type 1 diabetes mellitus. That means it helped to associate genetic relation and disease. Where there is the presence of HLA DR3 or DR4 type of Mhcs, we can say, then there will be the association of occurrence of diabetes mellitus type 1. And where there is the presence of HLA B27 type, then it will be associated with ankylosing spondylitis. What I said is ankylosing spondylitis. Okay, that is the genetic association of disease with MHC. Now talking about another important uses of it is, it helps in organ transplantation. Okay, talking about organ transplantation, while transferring organs, we're transplanting organs, the thing that we should concern about is we need to be sure that there is HLA matching, or we can say it gene matching. Okay, that means for organ transplantation we should perform HLA matching, and this can be done by gene matching by tissue typing. And how is this process performed? This is performed when done with mixed leukocyte reaction and microcytotoxic assay. What I said is, this is done by, how it is done? It is done by mixed leukocyte reaction, and another thing is, another process is microcytotoxicity. Okay, that is what we need to know about MHC, major histocompatibility complex. That is what we need to know, and this is all about MHCs.